Guys, we will discuss about some basic techniques in genetic engineering. So what are the basic techniques which are utilized in genetic engineering? These are agarose gel electrophoresis. Then there are another agarose gel electrophoresis, then isolation and purification of nucleic acid, how they are isolated, then isolation of chromosome. Okay, isolation of chromosomes, nucleic acid blotting techniques, DNA sequencing, alternate methods of DNA sequencing, chemical synthesis of DNA, methods of gene transfer, polymerase chain reaction, production of monoclonal antibodies, construction of gene library, and radio labeling of nucleic acid. These are very, very important for the lab experiments. So let's start with agarose gel electrophoresis. So what is agarose gel electrophoresis? First of all, what is electrophoresis? So it is the movement of charged molecules in the electric field, and the negative charged molecules, they move towards the positive electrode, while the positive charged molecules move towards the negative electrode. Okay, it is used for the separation or purification of specific DNA fragments. Okay, and the gel is made up of polyacrylamide or agarose gel, and this polyacrylamide gel is known as page. Okay, in short we say it as page, which is used for separation of smaller DNA fragments. Okay, and the smaller DNA fragments will migrate further. Okay, if this is a gel picture, I'll show you, will migrate at to the far distance, while the bigger ones remain to the top. And here it is used for separation of DNA fragments ranging from size 100 base pair to 20 kb base pairs, 20 kb pairs. Okay, it can be also used to separate RNA molecules. So DNA as well as RNA molecules can be separated. Okay, now this is electrophoresis. What is agarose? Agarose, it is a polysaccharide, which is a kind of sugar derived from seaweeds. It's a solid gel when it is dissolved in aqueous solution. Here aqueous solution, what we use some kind of buffer. So at concentration between 0.5 to 2%, there are different percentage of buffer or different percentage of the solution we use to separate different kinds of fragment. We will discuss about it. Okay, and it is used for electrophoresis in a more purified form of agar. Okay, then we come to how this process is performed. So DNA samples are placed in the wells. There are wells into which the DNA samples are placed. Okay, wells are made by using the caster and comb. I'll show you the picture. And the power supply is then switched on. Okay, since DNAs are negatively charged, it will move towards the positive side of the electrode. As you can see here, these are the wells. Okay, these are the wells where the DNA will be like put on. This is the cover, this is a power supply. Okay, here this is a negative electrode and positive electrode. The gel is there, this is the made up of gel, this is the gel where the wells are there. Okay, and into the wells this DNA samples are loaded. Okay, DNA samples are loaded. Okay, so DNA, what it will happen, that DNA will move through the gel when the power is on, and DNA, since they are negatively charged, it will move towards the positive electrode. Okay, and the rate of migration will depend on the size and shape of the DNA. If it's smaller, it will move farther. Okay, so smaller fragments will move faster than the larger ones. And gel electrophoresis, it can be used for separation of mixture of DNA fragments based on their size. Okay, so agarose, what it does, it forms pores. Okay, if you increase the amount of agarose while preparing the gel, the more the agarose, the smaller the pores will be. Okay, So it forms pores ranging from 100 to 300 nanometer in diameter. And the actual pore size, it will depend upon the concentration of agarose, as I told. Okay, the size of the pores will determine the range of DNA fragments that can be separated. If the fragments are very, very small, we prepare a higher percentage of agarose gel. For instance, for 0.3% agarose, okay, it is, since this is a very less amount of agarose, it is used for separation of smaller, okay, less agarose, it will be used for the separation of bigger fragments. Less agarose, it will separate what? Bigger fragments. Okay, and a 5% agarose, when it is more, it is used for smaller fragment, 100 to 500 base pair. Okay, when agarose concentration is more, the pore size will be even more smaller. It will separate the smaller fragment. Okay, and the migration of DNA fragments, it can be monitored by using dyes. Since DNA are colorless, we have to put dyes before loading it into the well. Okay, to know that it has migrated a certain distance or not. Okay, if you will not switch off the power supply after a certain period of time, then all the DNA sample will be washed off. So we have to take care, and we have to monitor that how much the dyes have, it has passed, it has traveled through the gel. Okay, so to see where my fragments have moved till now, we have to put the dyes into the DNA. Okay, and dyes into the DNA as well. And to see the dye, to see the DNA, okay, to see the DNA, we put ethidium bromide in the gel. Okay, so there is a dye which we put into the DNA, and there is another dye which we put into the gel while preparing the gel. To visualize the DNA, we have to put ethidium bromide into the this agarose solution while we are preparing it. Okay, so the bands of the DNA, it can be detected by ethidium bromide solution. Okay, and this, when we prepare the gel, okay, we add agarose plus buffer, then we cool it down a little bit, then we add ethidium bromide, ETBr, then we pour it into the caster. This is the caster, and then we pour it here. Okay, and then in the DNA sample we mix DNA plus a certain kind of dye, that is bromophenol blue, to visualize the DNA, like how much our band have moved. Then into this band, say for example my band has moved up to here. Okay, and to see the DNA in this band, okay, this is the color which is seen is of dye, but to see the DNA in the dye, we have to put ETBr in the gel, which we, when we put ETBr in the gel at the time of pouring of the gel into the caster. Okay, so ETBr is what? When activated by UV light. So when the band have moved forward, when the band has traveled to the gel, we visualize it into the UV trans illuminator. Okay, okay, what it does, it it emits UV radiation, which is then absorbed by ETBr. So DNA base pairs in association with ethidium bromide, they emit orange fluorescence. You will see that orange color band in presence of UV light. Okay, so the separated bands can be identified by this gel dop or UV trans illuminator. Okay, we can visualize the orange colored bands, which are like colored orange due to the presence of ETBr in presence of UV light. Then there is another kind of gel electrophoresis, which is pulse filled gel electrophoresis. Okay, so the large size DNA molecules, that is around 10 megabase pair, it can be separated in agarose gel by using page. by using pulse filled gel electrophoresis. Okay, how it is done? By periodically altering the direction of the DNA migration. Okay, by changing the orientation of electric field with respect to the gel. How it is done? So when the field orientation is done, what happens? The DNA molecules align themselves and migrate in new directions. And there's a limitation of this pulse filled gel electrophoresis. It is that the samples do not migrate in straight line. So further analysis becomes difficult. This is a major drawback. Then another kind of gel electrophoresis, which is contour clamped homogeneous electrical field. Contour clamped homogeneous electric field. It is an improved method. Okay, improved method to separate DNA molecules. Here the reorientation angle is fixed, but here it is not fixed. Reorientation angle not fixed in pulse field. Here it is fixed. Okay, like for example 120 degree centigrade, okay, or even 90 degree, and electrophoresis is carried out. So large DNA fragments like 200 to 300 kb, they can be routinely separated in a matter of hours. Then comes PAGE, polyacrylamide gel electrophoresis. So what we've read, we read agarose, we read pulse filled, we read contour clamped. About polyacrylamide gel electrophoresis, it is composed of acrylamide monomers which are cross-linked with methylene bisacrylamide units. Okay, cross-linked with methylene bisacrylamide units. Okay, and the pore size is dependent on the concentration of the monomers as it is in agarose. And the cross-link is also dependent on the concentration of the monomer. So what is the monomer made up of? Acrylamide, chains of acrylamide, which is cross-linked with methylene bisacrylamide units. So this PAGE, it is used for separation of single-stranded DNA molecules which are different in their length by just one nucleotide. So can you imagine how much sensitive it is? Difference in length by just one nucleotide. So agarose cannot be used for this purpose. Okay, why? What speciality do this polyacrylamide gel have? Because they have smaller pore sizes than agarose gel, even smaller pore sizes than agarose gel, and they allow precise separation of DNA molecules. That is from 10 to 1500 base pair. So it is a wonderful technique for fine separation of DNA molecules which differ from each other by 1 to 3 nucleotides. It is used in DNA sequencing, identification of amplified product from PCR. So there are other uses of gel electrophoresis as well. Okay, what are these? So it is also useful for understanding the molecular configuration of DNA molecules and protein nucleic acid interactions. Okay, and these are based on the principle that binding of a protein to DNA usually results in the reduction of electrophoretic mobility. So if we want to know that a protein is binding to our target DNA or not, then we will run the DNA as well as our like hypothetical sample where DNA is also binded. DNA is in contact with protein. So if it is in contact with protein, we will find that it will be larger in size because DNA is there with the protein. So it will be towards the upper side of the gel. But when DNA is alone, it will migrate faster because the size is smaller. So comparatively, by checking the size difference, we can know by like visualizing the electrophoretic mobility, we can understand if the DNA is alone or it is in contact with the protein. Okay, so this electrophoretic technique For nucleic acid interaction, it involves the addition of desired protein to double-stranded DNA fragments. So we'll add the desired protein to double-stranded DNA fragments, and this complex is then separated. Okay, and we can visualize it to understand the protein nucleic acid interaction. So that's all about gel electrophoresis. I hope it is understandable. So stay tuned. In the next video lecture, we will start about isolation and purification of nucleic acid. Thank you.